Okay, good afternoon, everyone. I'm Dr. Sheila Farrell, and I'm a medical officer in the Division of Rare Diseases and Medical Genetics at the Food and Drug Administration. I'm happy to be here today to discuss drug development and clinical trials. And this is a very basic talk, really just to give you some idea on clinical trials. It's not in 30 minutes, we can't get into real depth on this. So for anybody that might be from academics or pharma, this may be too basic for you, but the plan was to educate patients on clinical trials. As my team, part of my role is to review the data provided by sponsors or drug companies when they want to conduct a clinical trial in humans for the first time, and then also to review the results of the completed clinical trials when the sponsor wants to get approval to market a drug. I'm going to start by saying I have no conflicts of interest. I have nothing to disclose. And this talk reflects my views and should not necessarily be construed to represent the FDA's views. As far as an outline today, I'm going to briefly go through the stages of drug development, talking about discovery of drugs, drug development, the phases of drug development, including clinical trials. And then, because I know you were interested in potentially participating in clinical trials, I wanted to talk a little bit about the safeguards for patients in clinical trials and the safeguards for children in clinical trials. So the ideas that a drug or a compound might be useful in a disease, they have to come from someplace. Oftentimes, they'll come from new insights into diseases. So perhaps we understand that, researchers understand that a disease, something's missing from the body. So like a chemical is missing. So they may be able to design a drug that replaces that chemical and causes that disease to improve. Or they may know, say, that an enzyme isn't working that should work. So they design a drug that helps that enzyme to function so that, again, the disease can stop or improve. Additionally, sometimes drugs come from existing treatments that have unanticipated effects. And then we can use those drugs for other diseases. One example of that would be something like Rogaine. If most of you know, Rogaine is a hair regrowth drug. But originally, they tested that for ulcers in animal studies and found that it wasn't particularly helpful, but they did think that maybe it might have some effect on blood pressure. So then they did blood pressure trials. And during those trials, they noted that people were growing more hair. And that's where the idea to then do clinical trials on hair regrowth were, and now that's a marketed drug. So lots of places we can get drug ideas from, but from this phase, only a few drugs actually make it into the next stage. The process of drug development, researchers have to answer lots of questions about drugs. They have to answer questions about how is the drug absorbed? Once it gets into the body, where does it go? For instance, if we have a disease that's primarily involving the brain, we have to know, does it get into the brain? Does it cross the blood-brain barrier so that it can be helpful? How is it metabolized and how does it get back out of the body? What's the best way to give the drug? If we give it by mouth, does the digestive system degrade it so much that it's not helpful? Does it have to be given IV or subcutaneously? We have to learn what the benefits and the mechanisms of action of the drug are. We have to learn what the best dosage of the drug is, and that we can't just pull that out of thin air. There has to be some science behind that. We have to learn about the side effects or adverse events, often called toxicities associated with the drugs. We learn does this drug interact with other drugs? Are there certain drugs we can't give together? Does it affect the other treatments that we might be on? Does it affect different groups of people differently, such as by gender, race, age, ethnicity? And lastly, how does it compare to similar drugs that may be used for the disease that we're talking about if we're lucky enough to have another drug that works for this disease? So this is a simple way of looking at drug development, but it's actually a lot more complicated than this. But again, for a 30-minute talk, I'm just going to go and use a very simplified approach and say that there's a preclinical phase, phase 1 and phase 2 and phase 3. Preclinical is before patients get the drug, and phase 1, 2, and 3 are actually in patients. It's actually more complicated than this. There are phase 4 trials, there are phase 1, 2 trials, 2A, 2B, all kinds of permutations. But for the purposes of this talk, we're just going to focus on these 4 phases. And each phase is designed to answer certain questions. So the preclinical phase involves both tests that we do in the lab and tests we do in animals. And we're trying to generate important information about potential side effects and determine if the drug is safe enough to give to humans, or is it toxic and we can't give it to humans. If it is safe, we need to know what kind of side effects to monitor for in the clinical trials. Animal studies also allow investigators not only to look at the safety, but also to pick a safe dose to use in the clinical trials. Researchers give animals increasing doses of the drug and find out what doses cause side effects and what doses don't cause any side effects. And they can often use the doses that were safe in animals to calculate what the equivalent dose would be in humans, and that can be used for determining safe doses for the clinical trials. Once the preclinical phase is done, once they've done these tests and the sponsor or the drug company thinks they have enough information that they feel it would be safe to use in humans, then they have to submit an application to the Food and Drug Administration. The Food and Drug Administration has a team of people who look over all of this information and determine if they think that it's safe for this drug to be used in humans. Some of the things we look at are, how is the drug made? Who's making it? What's in it? How stable is it? So we have to know that the drug, because this isn't a drug that's already out there usually, that they're making it appropriately and it is what they think it is. They also submit detailed outlines for their planned clinical studies or study protocols. And we review that to be sure that people aren't being exposed to unnecessary risks. We look at the schedule of assessments that they have in there that tells us, when they're going to see patients and what they're going to do on those visits and what they're going to be looking at, what labs they're going to be drawing, to make sure that any side effects that we're worried about, that is in the protocol. We also get information about the investigators that are going to be participating in the trials to see if they have the knowledge and skill to run clinical trials. And the research sponsor must commit to getting informed consent from everyone in the clinical trial, and we'll discuss that at the end. They must also commit to having an institutional review board, which is an independent board. Oftentimes you'll see those at medical schools will have them, or hospitals, and this is a group of people who also look out for the safety of patients in the clinical trials. If the sponsor, the Food and Drug Administration, and the IRB agree that it is reasonably safe to start clinical trials in humans, then a phase 1 trial may start. Phase 1 trials are usually designed to primarily look at safety. They obtain information on how the body handles the drug, how the drug affects the body, what a safe dosage range might be, and also any side effects associated with increasing the doses. Usually phase 1 trials are done in healthy patients. Occasionally we will have to use them in patients with the disease, but usually for the sake of this conversation, it's healthy patients. It's usually a small number of patients, and it's usually for a short duration. So typically we'll start with a small number of patients with the lowest dose that we think that is potentially in the safety range, and we'll see how the patient does with that. And then if that dose is fine, then they'll try a higher dose. Sometimes they'll use the dose for a week duration to see over a week what happens. But they're monitoring these patients really, really carefully during all this time. Oftentimes they're staying at the research center overnight, potentially having their blood drawn, having EKGs looked at, all kinds of testing to make sure they're safe, and then they're reporting any side effects that they might have. If a safe dose is found in phase 1, then we need to know if the drug provides some benefit on some aspect of the disease. We want to know this before the sponsor puts large resources into a big study. And also, besides that, we're looking at side effects. We're giving the drug longer in a phase 2 than a phase 1, so we want to know if with time we're seeing more side effects. And about a third of drugs move on from this phase to a phase 3 trial, and that is if they found it to be safe and they believe it has some benefit. Phase 3 trials are large trials. They're larger than phase 2 trials, and they may have hundreds to thousands of patients in them. However, for rare diseases, say we have a disease in the United States that has 200 patients with the disease, we cannot expect to have 200 people in the clinical trials. So there is flexibility for smaller patient populations, flexibility in trial design and flexibility in the numbers of patients for rare diseases. These trials primarily need to show that there's a benefit, and in addition, they generate additional extra safety information. For most drugs, there will be 2 phase 3 trials to ensure that the results are consistent if they're concluding that the drug is effective. These trials have to demonstrate a response to a treatment with a drug on some outcome measure that is important for a patient, for how they feel or function. Occasionally, they will look at a biomarker, but a biomarker would be something like a blood level or an MRI or some kind of test that correlates with clinical benefit in a patient. So for instance, if they use a biomarker, say we use glucose, sugar level, if the drug decreased the glucose level from 251 to 250, they would have to demonstrate that that is a clinical benefit to a patient. Otherwise, we're typically using non-biomarker outcomes. And I'd like to just give you an example of one, just so you understand when I'm talking about an outcome, what I'm talking about. Say we have a patient that has a disorder that impairs their ability to walk distances. Then the outcome measure for the trial that determines whether the drug is effective, we might look at a timed walk test. So for instance, at the beginning of the trial, the patient would walk a certain distance that they're able to complete in, say, 6 minutes. There's something called the 6-minute walk test. And then they would start on the drug, and then during the trial, they would see how far they can walk on the same test, being done under the same circumstances at various endpoints. And at the end of the trial, we would see, did the patient stay the same? Did they get better? Did they get worse? That would be the kind of outcome that we would be looking at typically in a clinical trial. At this phase of development, generally the study has to compare a group of patients with the disease who are taking the drug with a group of patients with the disease who are not taking the drug. Because we need to know if we give the drug, would the patients have the same outcome if they didn't get the drug? If there is another drug being used for the disease already, then we can use that as the comparator. But if there isn't, then generally researchers use a placebo-controlled trial. A placebo-controlled trial generally has 2 groups of patients. One group gets the drug and the other group gets placebo. A placebo is an inactive substance that looks and tastes like the drug being tested, but has no effect on the disease. Everything else in the study should be the same between the 2 groups, so that any difference in the outcome can be attributed to the drug. And an important thing to remember is that experimental drugs are indeed experimental. That means the drug can have a positive effect, it could have no effect at all, or it could be detrimental. So you have to keep it in mind that a patient on placebo may actually be getting better treatment than someone on the active medication. We don't know. That's why we're doing a trial. I've stated that we want the 2 groups of patients to be treated as identical as possible beside getting the drug. So in these big trials, besides being placebo-controlled or active drug-controlled, we also often have them be double-blinded. A double-blinded trial is a trial where neither the researcher nor the patient knows who is getting the drug and who is getting placebo until the trial is over. A monitoring group not directly involved in the study does keep track of the group assignments. This ensures that researchers treat all patients the same way and that nobody finds out whether they're on the placebo or the drug inadvertently. This ensures that any benefit reported by the doctor or the patient is not the result of human bias that might affect the outcome of the trial. There is something called the placebo effect. You've probably heard of that before. And that's the tendency in all of us to feel better when we're taking a drug that we think can help us. The placebo effect can occur when the treatment is actually helping us, or when it is doing nothing, or when it is harming us. So that is why we want to keep the trial blinded if possible, so that none of those effects can affect the outcome of the trial. Another important way we can be sure the two groups in the study are the same is by something called randomization. Because patients with diseases can differ in the speed of progression of their illness, so some people may progress slowly while others unfortunately progress more rapidly, we want to be sure that the patients in both arms of the trial are very similar and that there's a mix, the same mix in both sides. So by randomly assigning patients, and that means if you were the patient, you can't pick that you want to be on the treatment or the placebo. Your doctor can't pick where they want to place you, but you're randomly assigned, usually using a computer model, to either the treatment or the placebo group. So you have an even chance of getting in either arm, generally. And that leads to the diversity being spread equally between the group. And this increases the chance that any benefit we see will actually be due to the treatment rather than differences in the patients in each group. About 25 to 30% of drugs move past this phase and on to potentially submitting an application for a new drug. For a drug to be approved in the United States, it must show substantial evidence of effectiveness. And usually the phase 3 clinical trials are what they use to show that. They submit an application with all the data from all these trials, and the Food and Drug Administration reviews it to determine if they think there is substantial evidence of efficacy, as well as if the benefit risk profile is favorable, meaning you're more likely to benefit from this drug than you are to be harmed from the drug. And that's all I have as far as the clinical trials go. Then I'm just going to talk a little bit about safeguards for patients in clinical trials. So we've already discussed that we have the drug company that's reviewing the safety. They're working with their experts to make sure that patients are safe. The Food and Drug Administration is looking at the initial trials and usually the protocols as they come in, if there are other protocols. The IRB is looking at this data. So we've got lots of people trying to ensure that the safety and the rights and welfare of humans participating in the trials are regarded. Other safeguards that are in clinical trials include informed consent, which we'll discuss in a minute, and then safety monitoring throughout the trial. And also some trials will have safety monitoring boards. Informed consent is required for enrollment into clinical trials. You should be made aware of potential benefits, potential risks, and what the study entails in terms of visits and procedures, and any risks for those procedures. It's really important that you not think of this as just a piece of paper to sign, but you should have all your questions answered before you sign a consent form. You should have sufficient time to review this without coercion. If you need to take it home, a copy of it home and review it with family, that should be fine. It should be in language that you can understand. If it's not something that you can understand, you need someone to explain it to you. They should explain it to you. If English is not your native language, there should be a translated version. The consent cannot ask you to waive your rights or release anyone from any liability. They cannot make unproven claims or certainty of benefit in the trials, particularly if this is a brand new drug. The reason we're doing this is research, right? So they have to have a statement there that says research, but they can't guarantee you that there will be benefit if it's a brand new drug. There should also be a statement in there saying the purpose of the study also includes evaluation of the safety of the drug. So while we might have some information from animal studies or earlier studies, we do not have all the safety information. And as you add more and more people, you're able to see more and more side effects sometimes if they're present, because you're giving it to more people. So things that occur in 1 in 100 patients, you're more likely to see in a large trial than you are in a small trial. The informed consent should also have a disclosure of appropriate alternate procedures or courses of treatment, if that may be advantageous to you, and also a statement on confidentiality. For research involving more than minimal risk, and minimal risk is usually, you go to the doctor, it's that kind of, you have your blood drawn, it's basic risks we take every day. An explanation as to whether any compensation, any medical treatments are available if an injury occurs, and if so, what they consist of, and who you should contact. So if there's more than minimal risk, they should be explaining to you what the consequences of that are and how you can address that. There should be a statement in there as well that this is voluntary and that you may withdraw participation at any time without penalty or loss of benefit if you decide to do that. As far as safety monitoring goes, while participating in the trial, the sponsor is monitoring for potential side effects based on either the animal data or the drug information they have from drugs that are related to this drug, or information from earlier trials. They're often doing multiple tests, which include blood tests, which may be looking at your liver function, your kidney function. They may be doing ECGs or echocardiograms. If there's indications for looking at your hearing or vision or any other things that they think might be possible side effects, they have a schedule for when they're going to look at those things throughout the trial. Maybe in the beginning, they're drawing your blood every week. And then as the trial progresses, they may be doing it, every month, and then every 3 months and 6 months. They are monitoring those things. And there is a safety board in some clinical trials, safety monitoring board, not all clinical trials, but usually there's somebody kind of looking out for the safety as the trial is going on. So they may review the data at that point, make sure that it's still safe for patients to stay in the trial, and maybe make some adjustments to the safety monitoring or even to the trial, depending on what they find. There will also be visits to the clinic, physician visits to monitor your health as well. And in this day and age, I think since COVID has come on the scene, we're seeing a lot more televisits and a lot more nurse visits out to homes as part of clinical trials. Sometimes they're drawing the blood, so it's limiting the number of visits you have to make into the clinic, but you still will have those clinic visits to have physical exams and for certain tests that can only be done at the clinic sites. The last thing I wanted to touch on was safeguards for children in clinical trials. Generally, in the past, very few trials were done in children. So doctors were really left without either not using a drug in children or using it off-label, having to come up with information based on what we know from adults. And a lot of regulations were passed that said that we need to try to label drugs for children if it is appropriate. Sometimes we can get the data to label the drug for children from the adult data, but oftentimes we can't, and we have to have trials in children. So there are a number of safeguards for children. Oftentimes we will have animal models that are juvenile animal models that will demonstrate the safety of a dose and the drug, so we can try to pick that initial dose for children. We can also use some of the data from the adults. We generally test drugs in adults first and then children. So usually we know that it's safe in adults before we allow children to have it, if that is appropriate. And there's some diseases where that may not be possible. We stagger enrollment. So oftentimes the older children, the 16 and 17-year-olds, will be getting the drug before the younger children. So we'll get some data about dosing and safety from those children. And we do have rules that children shouldn't be subjected to any testing that isn't necessary. So you have to be thinking carefully about what tests are necessary for children. And there are additional safeguards in the regulations, and it's called 21 CFR 50 subpart D for anybody that might be interested in reading anything about safeguards for children. Generally, if someone has not reached the age of consent to treatments or procedures involved in a clinical trial, then a parent or guardian must consent for the child to participate in the clinical trial. Assent from children who are old enough to understand what the trial, obviously they're not going to have the adult understanding, but just be able to understand what's going on. Someone will explain it to them, and we try to get assent from children that are old enough as well. But for research in adults in general, the adult subject does not need to necessarily benefit from the research study. In adults, a research study is generally okay if the risks to the adult subjects are reasonable and the research will generate important information. So for instance, when we talked about those phase 1 clinical trials, those are healthy adult volunteers usually. They really are generally not going to benefit from participating in the trial, but they are allowed to participate in the trial because they can consent and the risks are felt to be reasonable. However, for research in children, the acceptable research risks are limited, especially if the research is only gathering general information. For any research study in which a child may be exposed to risks to their health, the child must be able to benefit from enrollment in the research study. And what we would say is that there needs to be the prospect of direct clinical benefit for the child. That can be determined from many ways. It could be could be from adult data that we have or animal data that we have, but there has to be some anticipation that the child, that individual child, will get something out of this trial. And that is, make sure, yeah, that is all I have for you today. I'd like to thank everybody for listening. I appreciate the chance to come speak to you about this important decision that you're making. And I'm happy to try to answer any questions that you might have.
Awesome. Thank you so much. So we had 6 questions that were submitted ahead of time. And anyone in the audience who has questions right now, just submit them to the Q&A and we'll try to get to those. So Sheila, there were, all right, so I guess I'll just start at the top of the list. One question was, what are some of the requirements for an N of 1 trial to be able to take place and the typical timeline with that?
Yeah, I honestly cannot comment too much on an N of 1 trial. I have not had to deal with one of those yet. I do know there is probably guidance on that. If you go to the FDA website,
okay,
you should be able to look and see if we have guidance on that. And if you can, I can get you some information on that, Celeste, and send it to you. If you know who the person is that would like that information, or if you want to send it out in general, I'll get you some information. That's a newer thing, and I've read about it, but I don't actually know enough to speak competently about it.
Perfect. Let's see. Another question was, upon success of gene therapy, do you anticipate any age limits of children to get the gene therapy?
Is that specific to a certain disease, or?
I think it's in general.
Yeah, I think for gene therapy, it would really depend on the disease and whether or not they think that it would be efficacious if you give the gene therapy later. So that would really have to be disease specific in terms of, and that would be something you would have to discuss with you could discuss with the doctors involved in the clinical trials.
Of course. Okay. The next one is there a huge difference between clinical trials of drugs versus devices versus biologics?
Well, the standard is the same as far as substantial evidence of efficacy. The way the trial might run might be different. I mean, even within clinical trials for drugs, we can talk about these general comments that I've made, but there are nuances to everything. So there are nuances to devices, there are nuances to drugs, but the overriding principle is the same: is that you have to have a clinical trial that demonstrates that your product, your drug, is substantially efficacious and that the benefit of it outweighs the risk.
Okay. The next one is, so I know one concern from our community, because we are such a small community, and I'm sure you see this across the board with rare diseases and ultra rare diseases, but is it possible to not use a placebo in a trial because of the small population?
That's a great question. And yes, we do see this all the time. And the the standard is definitely placebo-controlled trials. We feel that it is the best way to get the information. But if the natural history of the disease is known, so for instance, registries, if your organization has a registry where they've been collecting data on patients, which many diseases do, and NORD has a lot of information on that, then you can, if you can demonstrate that we know about the natural history of the disease on a certain aspect of the disease, and some of these registries, they'll collect data. So say we talked about that walk test that we were talking about. And this is just a general example. But say you've got a disease for which patients have been, they're in a registry and they're getting some type of test every 6 months over the years, and we have that data. Sometimes you can compare the results of your trial to the patients in a registry. But you have to, there's a lot of nuances to that as well in terms of, that it be the same outcome, and how was the data collected and all that. So it's complicated, but it definitely is possible if you have good data on the natural history of the disease.
That's perfect, because we actually have a registry that we have launched through the NORD platform, and so that is just more oomph behind our drive.
Absolutely.
Yeah. And data.
Participate.
Yes, participation. There's another question. What can we as parents do to support drug development?
Well, I think that I think belonging to organizations, like NORD and this organization that you're belonging to, where you're with a group of people that are kind of looking for the same types of outcomes. You definitely can talk to your physicians. I'm assuming most people are in specialty clinics, where you're dealing with physicians that are experts in these diseases. So talking to them about what's going on so that you're aware of it. You can look, as far as clinical trials, there's clinicaltrials.gov to keep yourself informed about what might be out there. So I think just being involved and talking to your doctors about what's going on, and what's making sure that you're up to date on the information, the latest information that we have on everything. I think if you're here at this conference, you're doing the right thing for your child already by participating in a group.
Yes. And then the only other one that I have, another one just popped up. We covered placebos. It says, is it possible to have trials that don't involve placebos? We talked about that, especially if there's early results that are promising. So yes, Dr. Farrell was saying that it is possible, and it helps to have data to help inform that.
Yeah, and the other thing I would say on that, Celeste, is, if the outcome is something dramatic, and I'm trying to use something that, that you, like, say we know in a disease that, at 5 years, no one's kidneys are working, say for a disease that maybe causes the kidneys, then that's natural history data, right? That's what we know about the natural history of the disease. So or let's make it a shorter time period, let's say 2 years. So then we know if we do a clinical trial and at 2 years, we have half the patient's kidneys are working, when we know that from the disease, from good data that we have, that no one normally would have their kidneys working at 2 years, then that is something where you have a dramatic effect. So when this person was asking, if we know it's promising, that's the kind of thing that we would be talking about as promising. It's something that has a dramatic effect, that no one can really say that this patient would have done this if they weren't on the drug, because we know either from our registry or from the natural history that this would never have happened without a drug. Does that make sense?
It does. And there's a follow-up question that just popped up. How long do family history studies need to be conducted before, or it says before things are allowed to progress to a clinical trial? So I'm thinking maybe they mean, like, is there a certain time frame that you want to see data from the registries
Yeah, I understand. Yeah. Well, you need to collect it from some time, and a lot of times it would have to do with the outcome that you're looking at, right? So, for instance, if you were going to compare patients in a trial where they were getting just the drug over, say, 2 to 3 years, you'd have to have patients, in the same age range who were being monitored with the same way over those years. Or if there was an outcome that you're looking at, you'd have to have a period of time of observation if the drug isn't something that works in 6 months. You know what I mean? If it's not like a huge change, like what we said, a dramatic change in a short period of time, you're going to need to have a reasonable amount of time, because that's how you learn about natural history of a disease, right? Is by following it over some period of time. So I can't be specific about it because it would really depend on the outcome. There may be some outcomes that you would have information on in a short period of time, but in slowly progressive diseases, it's going to be a longer period of time.
Gotcha. Yeah. And we have the registry, but then we also have a natural history study going on as well. So hopefully those are good things in our bucket.
Yes.
Someone else just asked, can the targeted outcomes change over the course of the trial?
That's a complicated question, but I would say generally the outcomes need to be what we call pre-specified. So usually what happens is there's the protocol we kind of talked about, the plan for the study. In that study, they pre-specify what the primary outcome is going to be. So what's the main thing we think this drug is going to do? Do we think this drug is going to, improve glomerular filtration rate? Do we think it's going to allow somebody to walk farther? That is the primary outcome of the test. And then there's a bunch of secondary outcomes. So those usually are specified ahead of time. It becomes, and it's very complicated to explain, but it becomes very problematic if once the trial has started, you start changing things. It's usually, we like things to start with a plan. And now you may discover things during the course of a trial that lead to other, investigations. But typically for a trial, we like to have things pre-specified on outcomes.
Okay. That makes sense. There was another question that came through. Is the family history study required before FDA is willing to consider anything? And I don't think it's required, right? It's just good extra data to have on the table.
Yes, absolutely. Yeah. The more data that you have, the better off you are. And like I said, if there is, when we talked about just placebo-controlled studies, but there are external control studies that use data that's already out there. And so for those types of studies, you need to have more information. But information is power for any of these, for any trial, really.
Yeah.
To know what the history of the disease is very helpful.
Yes. Well, that's all the questions that I have right now, other than, I know that you work in the rare disease space, and you've mentioned there's a lot of flexibilities for rare diseases in regards to clinical trials. So are there any other flexibilities that you can speak to in regards to clinical trials?
Well, I think, I don't know whether it's flexibilities, but I guess one of the important points that I would just make for a patient group is that if you're aware of clinical trials that are starting, early in the early phases, it's really important for you to have a voice with the company, the investigators, to tell them what outcomes are important for you. And Celeste and I just briefly mentioned that, but we need, for a drug to be approved, we need to know that it's clinically meaningful to the patients. And so a cure is obviously what we would all want for any disease, right? That's the goal. But there are other endpoints that might be meaningful, even if it's not that. So for instance, if I have a disease where my energy is really low, even if you can't cure my disease, but you can create a drug that gives me more energy, that would be meaningful to me, right? And we know that from patient groups, that's one of the things, and for certain diseases, that they just want to be able to get through the tasks for the day. So I think it's really important that you are informing as patients and parents and patients the choices that are being considered in trials for outcomes, because that's what we're looking at when we're looking at the trials. We're saying, is this meaningful to patients? So starting early and having they can do interviews and all kinds of things to try to get the information about what would be a meaningful outcome for your child or for yourself.
Absolutely. And as caregivers too, I know with my son, we'll be caring for him for the rest of his life. So,
Right.
what would we like to see for our son? So,
Absolutely.
and a question popped up: can the method of administering a drug change after the drug is approved?
Yes, it can. I'm not exactly sure where you're going with that question, but sometimes they might start out with the drug that they're giving, typically like maybe giving intravenously, and then they decide maybe we can do this like under the skin subcutaneously. So that can happen. They can come up with other forms of the drug. Now they usually, to change that, they often have to submit data to the Food and Drug Administration. So say it gets approved for an intravenous form, but they want to do it subcutaneously, we'd have to change the label. And so oftentimes we would have to get information that says that it's the same level of effectiveness and that this is safe for patients, et cetera. So, but yes, the form can change. Did I answer that question?
I think so.
Okay.
All right. Well, I don't see any other questions that have popped up. I can't think of any additional ones. So on behalf of the Association for Creatine Deficiencies, I would really like to thank you for your time today, Dr. Farrell. And then I will follow up with you to get just some information that we spoke about. And then so
and I will get you that N of 1 information.
Yes, that's what I was referencing. So, and thanks everybody for participating. We'll have a link for this on our website as well as on our YouTube channel. So have a great weekend.
Thank you so much. Best of luck to everyone.